Okay, so thank you for the introduction. Again, I'm Eva, and even though this is a Python video, I will not actually be talking so much about Python, but more about the story behind the research I'm dealing with, which deals with native proteins. Proteins, those tiny things that are in all bodies that makes the bodies work. If I give you an example of hemoglobin, that's something you all probably know, that's the protein that goes from our lungs to all other parts of the body and carries that oxygen, so it helps us breathe. Proteins are quite important for us, and it's pretty fascinating to study them. So let's have a look how protein looks like. You can imagine it as a long string of tiny beads, where the bead is called an amino acid and is the building block of the protein. We have 20 different types of amino acids. You can imagine it as 20 different colors of the beads. And from this, you're building the whole protein. As long as the protein is finished, it has its full length, it does not stay in this linear form. It immediately falls into some 3D structure. And this 3D structure is very important for the protein because it determines its function. So, for example, this orange thing, it looks like pocket, for example, and its function might be that some other molecule sticks to this pocket. Or the blue thing, it looks like a channel, so it sits somewhere in a cell membrane and it guards which things can slip through in and out from the cell. If we have a look at the folding itself, if the protein is somewhere naturally in a cell, it happens like immediately, it's in milliseconds. But when you're a scientist and you have this amino acid sequence, which is quite easy to get with some experimental methods, how long do you think it will take you to get the 3D structure? And you want the 3D structure because there's the function of the protein, that's what you're interested in. So let's do something like a warm-up activity for you. How long do you think it will take a scientist to get the 3D structure of a protein? We can do like measurement with our hands, like
arbitrary 3D structure, or any 3D structure, because you know one of those things sounds way easier than the other.
Okay, arbitrary 3D structure of some protein.
Alright, I'm a physicist from totally unrelated field, so I would guess like days in crystallography or weeks in crystallography in mathematics, and unfeasible today by computation methods. Right? Mm-hmm. Well,
the first part is kind of true, but not really, because with the experiments it can take you more like months if you know what you're doing, and if not, it's more like in years. So there's a joke about this. It's not a nice joke, but it's in the scientific community that every protein 3D structure that is somewhere in a database took a PhD student his life. So it's not like this is not really nice. And this gap between the protein sequences and 3D structures is really nicely demonstrated in the size of databases we have with proteins. So, for example, UniRef, it's a database of all protein sequences. It is something like 300 million sequences there. When you want to work with those sequences, you want to cluster it, so you're at something like 50 millions of protein sequences. But if you want to have the 3D structure, it's something like 190,000. So there's like a huge gap between those things. And this plot quite nicely shows that this will definitely not get better in the future years. So you probably need something like a computational tool that would help you to get from the protein sequence to the 3D structure, right? And luckily we have a cool thing for this. It's called a CASP competition, which is like a group of people who every 2 years publish some protein sequences with unknown 3D structure. And basically anybody can join this competition and try to predict the 3D structure. And then the guys come and get a real structure and compare it with the model 3D structures, and you can see like who does the predictions the best. Every year the results look something like this. You have some models on the x-axis who join the competition, and then the score on y-axis, how well you're doing the prediction. And then year 2020 came, and there was AlphaFold who like completely rolled over all the other tools. And it was a pretty big thing, because this tool was able to predict the 3D structure with almost the same precision as the experimental methods. So That's a big thing. Now, what's AlphaFold? It's not surprisingly a machine learning thing, and it's trained on all three structures that were available at the time. You can see the schema what's inside, and it can be split into like three parts. The first one deals with encoding the protein sequence, and actually I didn't know one thing about proteins, which is pretty fascinating, and that's if you have two protein sequences that look kind of the same, it's very probable that their 3D structure will be almost the same. So we have here an example of human, mouse and fish hemoglobin, and if you have a look at the sequence from human and fish, there's maybe like 50% of the sequence the same, but the 3D structure looks almost the same. That's because the function of this protein is the same, right? It carries oxygen. So if we jump back to this schema, that's exactly what's happening in this first part. You have your input sequence, and you're doing something like database search for something that would be similar to this sequence. And what you hope for is that you can find maybe some patterns there which would help you with the 3D structure prediction. Then there's another input, which is like modeling potential contacts within the protein. And the last logical thing, you're looking for some already existing 3D structure that will help you with the prediction. The second block is doing like inside magic there. There's the word evoformer. It sounds a bit like transformer, right? And that's exactly, not exactly, but almost what's in there. And the last part is like decoding from the inner representation to the real 3D structure. So it's outputting like XYZ coordinates of the all amino acids. If you want to predict your new protein, the 3D structure, it's not so simple because this whole thing is quite big, it's computationally extensive, so it takes maybe hours to run it. This is like great because we don't have to do the experiments which took us years, we can do it in an hour, but still, can we do it somehow better maybe? So this part, this database search for similar proteins, is a slow part of this thing. So the idea is, what if we try to replace this thing with some protein language model that kind of understands the proteins, maybe in the same way as the database understands them? And here comes the ASM-fold. That's a tool that basically copied AlphaFold's architecture, but just replaced this slow part database search with faster language model, and they're able to do the predictions almost in the same precision as AlphaFold, like they're really good, and they're doing it in like seconds. This was a bit long introduction about proteins and their 3D structures, so let's jump now into large language models. What is that? Large language models are, in simple words, type of an AI that can kind of understand and potentially generate some text. They nowadays usually trained in a specific manner. They use in a big database, for example, whole Wikipedia articles, and they train the way that you give them some sentence. For example, there was a king who had 12 beautiful daughters, with the word daughters masked, so the model can't see it. And what you want from this model is to predict you the probability distribution of this hidden word. So it outputs you a couple of words with some probabilities, and here it outputs the word daughters with the highest probability. So that's exactly what you want from this model. And however, training these types of model is quite hard because they really big and big data set for that. So usually only big companies can afford this. But when the model is already trained, it can kind of understand the language, and you can then use it for some subsequent tasks. So, for example, classical thing, when you have a movie review database with positive and negative reviews, and what you can do is just download some of those language models. You cut the last layer, which is predicting the next word, and you replace it with your own layer, which is like outputting positive or negative, and you fine-tune this thing on your dataset. And what you hope for is that the model already kind of understands the text you're given it, so you only slightly adjust it and it outputs you what you want. And this fine-tuning has become really easy today because we have, for example, Hugging Face. So for those of you who don't know Hugging Face, it's a really cool thing. It's like a platform for data scientists and machine learning, where you can easily share models and datasets. And if some model is available for Hugging Face, it's really easy to fine-tune it on a dataset. It's like 10 lines of code where you just load the dataset and you train the model, and that's it. Now, I was talking in the past couple of minutes about natural language. How does this relate to proteins? It's actually quite easy, because you can imagine proteins as some type of a language, and it fits there very well, because the amino acids, the building blocks of the protein, have even their own letter codes. So you can imagine that the amino acid is a word, and you can do the same thing as with natural language. You can mask this protein sequence and you can train some model on this. People have already tried it, so we have, for example, bird model, which is trained on like a huge database of all protein sequences. If you spotted this bird inside and you know what bird is, this bird has exactly bird's architecture, but it's just trained on proteins, not on normal language. And we have also, for example, ESM, which I've already mentioned in the previous part with the predicted tree structure. That's the fast tool. And we as scientists are really happy, because these models are available through Hugging Face, so not only us, but basically anybody can now play with proteins with the same way as I showed you, like those 10 lines of code. It's really easy. And I'm going to the last part, which is like the research I'm dealing with, which is about knotted proteins. I know it sounds a bit weird, but it's actually built on an old mathematical knot theory, which comes from like 18th century, which deals with categorizing different types of knot. So on this picture you can see like different types of knot you can have, from the simplest unknotted thing to 3-1 knot, which is something you probably tie on your shoelaces, and many, many other things. Just to explain here the terminology, the first letter is the number of crossings in a knot, and the second is just to distinguish two different types of knot with the same number of crossings. How does this work with proteins? It's the same thing. You're trying to see which type of knot the protein has, but it's not so easy, because you probably can't see for the first look if the protein is knotted or not. However, most of the proteins are unknotted, so if you pull the protein from both ends, it will stay in like a linear strand. But some of the proteins have a knot. It's like about 1% of the proteins, and the most common knot, 3-1, is like the simplest one. But we can have other beautiful guys like 6-1 knot, or even like double 3-1 knot. Even though knots have been studied for quite some time, we're still not completely sure what's the purpose of knot in the protein. There are some ideas that it, for example, helps the protein to prevent from degradation, or that we know that in some proteins the knot creates the active site, like the part of the protein which is very crucial for the function. And what we would really love to know, but we still don't, is if there are any amino acid patterns that are making the knot. And that's the place when we came up with our research. The idea is that we have a model that would be able to predict us if the protein is knotted or not just from the amino acid sequence. And then we can look inside the model and try to find maybe some patterns that might be responsible for the knotting. When you want to train a model, you need a data set. So one might say, okay, this is easy, you have this AlphaFold tool, it has a bunch of predictions, you can just look for knotted proteins, look for some unknotted proteins, mix it together, train model, simple task, easy, done. It's not that easy. If you would do it this randomly, you would end up with completely biased data set, and the model would load absolutely nothing but knotting. So you have to be careful about this. As in every machine learning project, it took us like 90% of the time to figure out the data set. So when you almost think that you're done, then somebody comes, you might forget about this small thing, and you're at the very beginning of the data set, so it's like, yeah, big process. So after like 6 months, we ended up with manually selecting protein families which have at least some protein with experimentally determined 3D structure that is knotted. So everybody will be like comfortable with those protein families. data set we got was, after all, quite nicely balanced and quite big, so it was like 200,000 sequences. It was pretty nice. And for the training, we used the Probert model, which I mentioned before. And even before the training itself, we were kind of curious, how much does the model already know about the knotted proteins? Because we know that these types of models can kind of understand the proteins. For example, if you have the embeddings of the proteins, like the in representation from the model, and you put them to 2D, you can see that the model kind of already understands, for example, from which kingdom the protein comes, or, for example, how the 3D structure of the protein looks like. So we tried to put in our data set and see how the model would work with that, and we saw, okay, it was pretty nice. The model kind of understands the knotting problem, even though it wasn't trained on that yet. So this was really good for us. We then took the Probert model, trained it on our data set, and got pretty nice results. It was actually a bit suspicious because the accuracy was around 98%, so it's quite a lot. So we were like afraid that the model might, for example, learn only some protein families, like the bigger ones, and totally forget about the small ones. So we checked this and everything looked like fine. So we then jumped into the interpretation part when we were trying to see if we can extract some patterns that would make the knot. We tried a couple of things, and the best showed up to be our own technique when we were like patching parts of the sequences, basically hiding it from the model and observing how much the model prediction for knotting would drop. So for each protein sequence, you then get part of it most important for the model's prediction that this protein is knotted. You can then pull all these things together and try to give them some biological meaning. So we tried this for one particular protein family, and we observed that the model is primarily focusing at like the very end of the knot, which is tightly coupled with the function of the protein, which was pretty interesting. And what we were also able to get was some pattern, which might be responsible for the knotting. But we did this only for one family, so it would be really nice to, in the future, to see this for other protein families. And like a bit crazy idea for the future is try to design our own protein with a knot. Now, you might ask, how is the whole thing useful? And it's actually a bit challenging question for us. But let me give you some examples. There's, for example, research that improper formation of a knot might be related to obesity, or we know that some knotted protein families are important players in our bodies, and that the knot is tightly coupled with a function. And one of their functions is that they are related to antibacterial resistance. So, for example, if we better understand the knot, we might be able to design better antibacterial drugs. So, to conclude this lesson, remember, proteins are cool, especially knotted proteins are cool, and the research on proteins is like really good. At the end, I would like to thank my whole team who worked with me on this knotted protein project, and also thank you for listening. And before we go to questions, if anybody has like any feedback for me to this presentation, you can come to me then after the talks and give it to me. I would be really glad for it. So, any questions?
So this 98% accuracy data, it was from the same data that is being trained on, or it's new data?
It's the same split.
Split it, so yeah.
If we tried it on like a new data set with different type of knot, it wasn't so good, but it was like a different type of knot which it has never seen. So we are not sure how this thing works.
So just to clarify, you did do cross-validation, right? So yeah, that's fine. I mean, that was kind of the question, I guess. Okay.
But like it's better to have like a little bit of data that hasn't been presented in the training later.
Yeah, and that's what we did. It's like a split.
And how was the precision? You said accuracy was 99 and...
Sorry, don't remember, but like high. really high. Everything was high. It was, yeah, actually a bit suspicious, but it seems that the noting problem is kind of easy for the model to get. Okay, if no other questions.
Okay, thank you.